Welcome, everyone. It is Mr. Olson. Welcome to Biology B. Now, yesterday you should have learned learned a little bit about DNA. So we should learn about things like nucleotides, base pairing, hydrogen bonds, covalent bonds, all that good stuff. Now, today we're going to talk a little about the history. So, how this came along. How did we discover DNA? How do we know the structure? Things of that nature. So, it's not going to be too long, but let's get to it. So the first person we need to talk about is a man named Erwin Chargaff. So he discovered the idea of base pairing back in 1949. He was a chemist, and realistically, he discovered that A equals T and that C equals G. So adenine always is in the same, I want to say equivalence. I don't know if that's the right word, but it's always in the same ratio. There we go. Adenine's always in the same ratio as thymine, and cytosine is always in the same ratio as guanine. So A equals T, C equals G. It's not the structure of DNA perfectly, but it is an idea. Now, kind of hold on to us. Erwin Chargaff did this in 1949. You guys do not need to know all the dates, but I do want to remind you that we're going to kind of do this in like some type of chronological order. So I'm just having the dates there just to help you out a little bit. Now, the next people we need to talk about is a woman named Rosalind Franklin and another man named Maurice Wilkins. So Rosalind Franklin and Maurice Wilkins did X-ray crystallography. And realistically, what this means from my understanding is that they take X-ray photos with light, and this light refracts images or refracts shapes, and it gives you an image. Now, Rosalind Franklin's most famous photo was this one on the right-hand side over here, and it looks like an X. This picture revealed a helix shape, so it was taken back in 1952. Now, if you guys remember, a helix shape's that spun up ladder, so it's like spins upwards. If you looked at that straight downwards, you would see an X because that spiral shape actually does create an X when you're looking at a 2D plane straight up and down. Now, this is huge. This is the first time that this image was taken and the first time that someone really has an idea what this structure is going to look like. But here's the catch. There's another two men named James Watson and Francis Crick that were actually looking for the structure of DNA. So were Rosalind Franklin and Maurice Wilkins, but James Watson and Francis Crick were as well. Now, they were kind of those dudes that just kind of gathered information, collected other people's information, and kind of just did their own thing. They were known to like go on walks and just talk about DNA and try to like figure it out that way. I wouldn't say they were lazy, but they weren't as straightforward as, say, Rosalind Franklin was. Now, they made models, and their models are usually made with minimal data, so they just kind of put stuff together. But they did see the X-ray photo, thus taking of the helix shape. So this photo right here, they saw, and they actually weren't supposed to see it, and they took this image without permission, and they used it. So when they had this image, they took Chargaff's rule of A equals T and C equals G. They took Rosalind Franklin's image, and they actually were able to go back to the lab, build a model, and they built the first correct model in 1953. Okay, now this sounds great. They got to where they're going. Now, Maurice Wilkins and Rosalind Franklin were also trying to discover the structure of DNA. James Watson and Francis Crick were also still trying to figure out the images of DNA or what it looked like, but they weren't sharing each other's information. So James Watson and Francis Crick kind of went under the table and kind of did it dirty to get this image. Now, what happened is that James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin, they all published their findings in an issue of Nature, which is kind of like a science article or a science journal. In 1962, James Watson, Francis Crick, and Maurice Wilkins, they won the Nobel Prize of Physiology of Medicine for the discovery of DNA structure. So they first built this structure in 1953, and it was actually awarded a Nobel Prize in 1962. Now, here's the negative part about this. Rosalind Franklin did die of cancer in 1958, and she did not receive recognition like the other three received. You cannot win the Nobel Prize when you are dead. It's just the truth. You have to be alive to receive the Nobel Prize. Now, here's kind of the messed up part. Rosalind Franklin, a lot of times, if you Google who discovered DNA, you are going to see James Watson and Francis Crick. Okay, Watson and Crick are always going to get the credit for who actually discovered the DNA molecule or DNA structure. And we all know now that's not fully the truth. So I just want to bring to light that Rosalind Franklin, this woman who did crystallography, this woman who was a scientist, deserves some credit. Now, did she die? Yes. Did she do a lot of amazing things in the science world? Yeah, look it up. Okay. She did a lot of really cool things, but she did end up dying of cancer in 1958, which is only 5 years later after the DNA structure actually fully came out. Does she deserve to have a Nobel Prize? Yes. But can she have one since she's deceased? No. Now, this is just kind of putting everything in something out there so you guys understand. But I want to make sure you guys got Chargaff, which has a Chargaff rule. So A equals T, C equals G. Rosalind Franklin, Maurice Wilkins. So talking about crystallography a little bit. James Watson and Francis Crick. And then kind of just tying it all together. Now, after this, guys, you're going to have a reading most likely about, I think, DNA replication. So make sure you guys get that done. If you guys have any questions, please reach out to me or any of your other teachers. Other than that, everyone, have a great day. Hope to see you guys soon.